when we transplant allogenic, so from one person into another, we get immune recognition, activation, rejection. So we spent many years understanding how that happens, which immune populations are responding, how the immune system corresponds with each other to lead to that rejection. And what we have found is that the first step is the HLAs, the fingerprint of the cells, are recognized as 4-ion T cells are getting activated. And T cells are part of the adaptive immunity. That means that part of the immune system sees an antigen and gets educated. So when you now retransplant, for example, those cells already know that antigen and they can react faster, more potent. So they are learning, it's adaptive. And what those T cells are doing, they can either directly reject the graft, and they are also activating B cells that antibodies get produced, and that you also observe antibody-mediated rejection. So to overcome that, we were looking into nature. We were very interested in understanding the concept where allogenic transplantation is working, and that is during pregnancy. It's not a transplant setting, but the fetus during pregnancy is 50% from the father. So 50% of the proteins are not known by the mother's immune system, but the immune system is still not rejecting the fetus. And we know now that the mother's immune system is recognizing the fetus. So it sees the fetus as 4-ion, but it doesn't get activated. So we spent many years in understanding that fetal-maternal tolerance concept, and we identified 12 to 13 molecules. They are either upregulated or downregulated by the fetus to avoid that rejection by the mother's immune system. And one very important concept here is that the HLAs on the cells, if you remove them completely, that this very first recognition by the adaptive immune system doesn't happen anymore. So what we did is, as a first step in the technology, we removed the fingerprint. We removed all HLAs. And we're doing that by knocking out the HLA class 1 and class 2 molecules with gene editing. And then you don't get this activation by the adaptive immune system. But then you face the second hurdle, and that means that innate immune cells, and those are NK cells and macrophages, they are in our body to sense if the fingerprint is missing. We call that the missing self. And the reason for that is if we have, for example, viral infections, the virus is knocking down those HLA molecules, and the innate cells are looking for this missing self, and they are killing virus-loaded cells because of that. So they are triggered to sense that missing self. So we had to overcome that hurdle. And we are doing that by overexpressing only one molecule that's called CD47. With that overexpression, we showed at the meeting that we can inhibit not only the macrophage killing, but also the NK cell killing. And by the combination of those three molecules, we are now overcoming adaptive and innate immunity. So that is the technology, and the rationale behind it is that we know from nature during pregnancy that the concept itself is working, and it was just very tricky to understand which molecules you need. And they were, of course, not allowed to modify the iPSC pluripotency or the ability to differentiate into different cell types. So at the ASGCT, for example, we showed now that we differentiated the cells into cardiomyocytes. And then we could not only transplant them, that they would survive allogenic in mice, but we showed that that they contribute to regeneration. So that after myocardial infarction, the heart showed regeneration after transplantation of the cells. We had shown another model where we showed we used endothelial cells, and again the hypoimmune endothelial cells did not get rejected, but we also could show that they are functional as endothelial cells. They formed new vessels, and when we had removed an artery to the leg in mice called peripheral artery disease in patients, those cells formed new premature vessels and rescued the blood flow to the leg. Another concept we showed at the meeting was the CAR T-cell strategy, because you can think about allogenic transplantation not only for regeneration. You can think about the oncology space, about allogenic CAR T-cells that then persist in patients without immunosuppression. And we showed some data there with human T cells, which were hypoimmunogenic in humanized mice, that they don't get recognized by the immune system, and that they're able to kill the tumor. In that case, in the meeting, it was a CD19 positive tumor, and then the CAR cassette was CD19 as well.